just throw something out there in terms of the aspect of, and quickly, you know, how can we help as scientists, either in the regulatory space, the industry space, the academic space, help build trust around NAMs as as we were looking for them to move to the future.
Press the button. Press the button.
We're on trauma right now. With Christy's group, we looked at where we thought we needed new approach methods and are changing it to sort of say questions that we would like answered and then put them on our website so that we can work with our partners through our research collaboration agreements, memorandums of understanding, CRADAs, that kind of stuff, so that we can work with all of you guys to help, because our budget situation anyway, but we can work with all of you guys to work together. So that's another thing that we're doing, is looking at where we think at the highest level, where we think we need some new approach methods, and then sort of translate them into what kind of questions, and then put them out there for along with Entree and hanging on that. So really good collaborative communities are also something that CRH has put together, where people just talk collaboratively, stakeholders and the government in areas like this too. So.
And Craig, did you ask a question? I don't know if you did ask a question or not, because if not, I will. And Wally, were you about to say something?
Yeah, I did ask a question, but
if you were alive in 1966, raise your hand. How many of, keep your hands up. How many of you remember the Animal Welfare Act of 1966 and what was in that Animal Welfare Act? You remember what it was?
Yeah.
We had to read it. So that's on that.
It states in that act that rats, mice, and birds are not animals.
Yeah.
So any of the numbers that I gave you related to reduction in animal use, how good they are, because NIH, this very senior North Carolina senator, got it put in that animals, rats, mice, and birds are not animals. Guinea pig may have been in it. I just remember those three.
And what I'll do is I'll see if there are any questions from the audience before we go to who's online. Any questions from the audience? And I can't see any questions online, so I don't.
So
No
we don't
questions
see anything
online?
here online either, but in terms of, I think what, I was asking is, and I think Susie started to answer was, what can we do from the various perspectives of industry, academia, and regulatory to help build trust in the system as we move to replace historically classic assays which we've relied upon. What we've learned is that when consumers lose trust, then bad things happen for all aspects of the continuum for keeping the food supply safe and plentiful.
And
Watch our website, because we're working on how you this Steve from Steve. Put him in. He's not here to defend himself. He's going to help us really look at the program entree that we put together and make it, and with comments from all of you, make it an even better program. So excited about that.
And I can chime in and just say, I think, some of the speakers in the first session, I think partnering people who have experience kind of communicating with, the general public. And, we're not always the best at doing that. I think that's one thing that we definitely need to be thinking about doing is how to engage with identifying people who can be champions for what we're trying to do, and be able to, express that and leverage that information to, the general public. I don't think we can do it by ourselves.
I have a website. I do have an email address. We're trying to get HFP NAMs that it goes to some people. We're trying to channel it to a few more people, but that's where people can write in and talk to us too. And this program NAMs.
I think we need more people like, and I'm not sure unbiased science is, that are willing to stand up and definitively say there's a difference between hazard and risk. And I think that's the thing that the agency has struggled with, particularly in the food area, because you're risk-based, but you don't emphasize the fact that exposure is the key to risk. And that just needs to be hammered on more and more. It's the toxicologist's fault. For the last 40 years, all we talked about was hazard. And only recently have we realized the mistake we made. We should have focused on exposure. And if there's no exposure or the exposure is so insignificant, does the hazard matter?
We have a question back here. Go ahead.
Just a question for Sharon.
Similarly, not to communicate, over promise what these NAMs will do. I think be very clear, or I get people, students and whatnot in my institution that really question, why does Europe ban certain chemicals and that the US is not? But similar with these NAMs, you need to communicate what they will and will not perform or tell you, because otherwise they're going to think the one assay in vitro is definitive. And you guys have done a really great job saying how it isn't. There's limitations.
Yeah.
And what might happen in one organ may not happen in the other, or consider digestion.
And I try to point out that those NAMs that are really effective, we know the mechanism. We know why they work. And that's going to be very difficult when we get into systemic effects. Because the public thinks cancer is cancer, but cancer is 100+ diseases, and they have different mechanisms. Different genes are involved, different polymorphisms.
You were going to say something, Sharon? I got a question back here, then we'll go here in the front.
So my question is for Sharon, and it's, your presentation was great. And I think this is part of the nutrition issue, because I think a lot of the NAMs and the development of these approaches is really looking for harm, whereas with human milk, you're looking for benefit as well as harm. So there's effectiveness and then there's safety. And so how do we do this in this complex dynamic system that is human milk that changes over time and is changing, with all kinds of environmental exposures? Are we able to even test whether the bioactive components will work in the matrix of, say, an infant formula? And then, of course, there's the safety issue. So that much further.
Yeah, thanks, Amanda. I mean, that's a great point, because I think that's the problem that we're having right now in this bioactive space, because really the FDA should be concerned about safety. But obviously parents and infant formula manufacturers are interested in efficacy. I mean, the main goal is to narrow the gap between breastfed and formula-fed infants. But how do you follow that? I mean, that's what that one workshop was saying. If you don't see, and these were immune experts, and Catherine, I know you were on that. If you're not seeing immune alterations by a year of age, there's probably little chance that you'll see things after that. because at least it started to narrow the scope, because right now, yeah. But see, again, there's questions, but how would a company support a 10-year long, post-market trial? So this is the thing. So it's that's why I said it kind of is blurring that line. And I know some of you may remember Eric Lean, and he was at Wyeth, and I always quote this, I think Paul, so he always said that, infant formula is the most tightly regulated food or loosely regulated drug. And it just straddles that space. So I'm not a toxicologist, but I slept at the Holiday Inn last night, but I've collected a lot of data that's been able to be used in grass applications. So this is a challenge, and I think it's very pertinent to NAMs, and my comment was really just, as we build out NAMs, can we look at ones that are reflective of pediatrics, when the brain is developing, or even a woman who's pregnant and her whole metabolism and blood flow and tissues are changed. I mean, could we have NAMs that would reflect different physiological states?
All right, we have time for just one more question because I know it is time for lunch. So, John, back here.
I just want to follow up on the toxicology aspect of hazards versus exposure. Because it's really broader than toxicology. We've chosen to apply that to nutrition. So we live in the world of using hazards to have good or excellent source of a whole bunch of nutrients, to be fat-free, to be sugar-free. So I'd like to hear your comments at moving out of just toxicology to bad things, but also how we've applied it within nutrients.
If nobody in the panel wants to grab that, I'll grab that in terms of it. It really is that aspect of trying to pin something on one constituent of the diet. So oftentimes there are forces who are pushing for dangerous or hazardous compounds, chemicals, whatever, where in reality, what we're really thinking about is the totality of the diet. So it's that mindset of, the hazard, as you said, driving down for claims that are supposedly helping to make better choices from a dietary perspective. So it is a slippery slope. And, the aspect of that's been brought up, at risk and benefit, is an important one where, we don't get the benefit of benefit in foods, even though much of the innovation in the food space in the last 20 years hasn't been, as Omari put forward, the classic food additives that have technical function in food, color, bulking, things like that. It's been aspects that are providing some type of benefit to the consumer to help make their diet better, really demonstrated by what Susan put forth around the bringing infant formulas closer to human breast milk. Throw it back to you guys.
All right, so I think that's it. You want to say something, Wally?
I'm just
Okay.
going to ask the audience, the Llewellyn clause says that if it's carcinogenic in an animal or human, FDA has to ban it, correct? The Llewellyn clause
No, it says.
for a food additive.
It says that
Food
FDA
additive.
cannot find it to be safe.
Safe.
So it's a very important distinction. It doesn't compel us to act. If we see something as a carcinogen, it prevents us from saying
That's-
something is safe.
Yeah.
With
That's the
question is brought,
important,
and this is very different.
that's the important thing, because the Surgeon General has just said that alcohol is carcinogenic, and I hope you don't ban it.
All right. Thanks,
So
everyone.
thank you, everybody. Thanks, everybody in the room. Enjoy the session. Have a great lunch. I wish I was there. I'll be there tomorrow. Hopefully I'll get to see everybody tomorrow, and talk more about this